Your memory of your first kiss, your emotion, your sense of personal identity and of your free will is nothing but behavioral of millions of molecules in a vast assembly of cells which we can call our brain. Francis Crick, co-discoverer of double helix, names this as the astonishing hypothesis. Astonishing to most of the people alive today who would like to use creative God or divine interventions or intelligent designer to explain the origin of such complexity. In contrast, comparative neurology and evolution suggest that the complex brain may be involved at least 5 or 7 times independently. And what I really would like to understand is molecular toolkits which underlie the very origins of neurons. A neurosystem seems to consist of several cell lineages which came together to form a complex brain and to perform integrative and signaling functions. So one of the major directions of my lab is try to identify the cell lineages and reconstruct ways how you can get a complex brain of octopus or honeybee or even a human. To do that, we try to use variety of the animals which have different level of neuronal complexity. One of them are ctenophores or cum jelly. These are representative of probably one of the earliest lineage of animal with true nervous and muscular system. Remarkably, these creatures have amazing regeneration capability. For instance, you can remove brain from one animal and in 3-4 days, 5 days it's regenerate. Then from the very the same animals you remove it once more and it regenerate again, and so on and so on. We do lost this ability, and I really would like to know how and why. Another example of model which we use in our lab is Aplysia californica. This cyst-lax leads the way to understand the ultimate mechanisms of learning and memory. And that happens because it has one of the largest neurons in the animal kingdom, only X are larger. And only 3 cells are sufficient to reconstruct a simple memory forming circuits in a brain, in a dish. So today, thanks to advantage of genomic technologies, we can monitor activity practically of all genes in individual neurons as they learn and remember. So in doing so, we address probably one of the most fundamental questions in modern biology: how activity of so many genes are integrated all together to form specific neuronal phenotypes, specific connections, and modify it to keep learning and memory. And I'm pretty sure Aplysia really will help us to understand why do we remember first kiss for the rest of our life.